Yes, students, in the previous class we have learnt about the process of fertilization, double fertilization and the post-fertilization events also. In the post-fertilization events, we have come across the main events such as the development of the endosperm, the development of the embryo. So now we are at this stage, we have to see the development of seed. So you know the technical meaning of seed. So technically, seed is nothing but a fertilized ovule. So you know that after fertilization, so within the ovary, see if you look at the gynoecium, so the gynoecium will be having the stigma, style and ovary. After fertilization, the entire part of the gynoecium, this lower part, that is the ovary, will be later converted into something else. The ovule present in this will get converted into, will mature into seed, and the entire ovary will mature into the fruit. So this is because of the action of different types of plant hormones released after the process of fertilization, after the syngamy takes place, right? So now we are talking about the seed. As I told you, seed is the fertilized ovule and it is the final part of the reproduction. It is the final reproductive part, right? And it is a very important part of the fertilization. It's a very important resultant part of the fertilization because it is this part which is actually responsible for the propagation, or you can say continuation of the progeny. So now this seed contains three main regions or three main parts. So the ovule, when it gets mature, the ovule after it is getting matured, so here this central part of the ovule, which is actually the embryo sac will become the actual seed and the integuments will become the seed coats. So here seed coats, the cotyledons, so it may be one cotyledon or cotyledons, more than one cotyledon, so depending upon whether it is a dicot or monocot plant, and then there will be an embryonal axis. So in the previous session we have studied about the embryo, the monocot and dicot embryo, where dicot embryo will be having a single cotyledon and monocot embryo will be having a single cotyledon, that is what is called as scutellum, whereas dicot embryo has got two cotyledons, where these two cotyledons later bulge up and store large amount of food material. So that is why, depending upon whether it is a dicot seed or monocot seed, one or two cotyledons can be present, and there will be an embryonal axis on which there will be the shoot apex and the root apex will be there, and in the case of the monocot seed there will be also epicotyle and hypocotyle. To protect the shoot apex there will be epicotyle, to protect the root apex there will be the hypocotyle. So like this, different regions will be there on the embryonal axis. So these are the three main regions, right? And there are different types of the seeds based on the endosperm, whether it is remaining or not in the during the development of the embryo. See, I have told you, in some seeds the embryo remains even after the complete development of the embryo. That means, so actually the endosperm is the part of the seed which actually provides the nutrition for the development of the embryo. But even after the completion of the development of the embryo, some part of the endosperm may be still remaining, and such seeds, they will be considered as endospermous seeds, or also called as albuminous seeds. So the seeds in which the endosperm is still retained or a residual endosperm is still present even after the complete development of the embryo, such seeds they are called as albuminous seeds. The other type of the seeds where there is no residual endosperm remaining after the development of the embryo, because during the development of the embryo, whichever the endosperm was there, entire endosperm, it is completely utilized. As a result of which, after the development of the embryo, there will be no residual endosperm remaining, and such seeds they are called as non-endospermous seeds, are also called as non-albuminous seeds. So non-albuminous means there is no endosperm present after the development of the embryo. Albuminous means there is the residual endosperm, remaining endosperm present even after the completion of complete development of the embryo. Example for non-albuminous seeds are pea and groundnut. So usually both of them belong to the family Fabaceae. Pea and groundnut, they still retain some amount of the endosperm after the complete development of the endosperm. Whereas you can see the examples such as wheat, maize, barley, sunflower, castor. In these cases, these are the albuminous seeds where residual endosperm, it is still present. Here it is not present, here it still remains, right? And these are called as albuminous seeds. And there are certain seeds. See, these albuminous seeds, after this remaining endosperm will be later used during the germination of the embryo. So that means because it was remained during the development of the embryo, it will be later utilized for the germination of the embryo or germination of the seed. So that means when a seed sprouts, there will be no more endosperm after the sprouting of the seed. Even in this case also, after sprouting of the seed, there will be no more endosperm remaining. But there are a few cases such as pepper, beet, etc. In some seeds, the endosperm remains throughout. Endosperm remains throughout even after the germination of the seed. So that means such endosperm, such persistent remnant of the nucellus. Okay, so endosperm or the nucellus, you know that. So here there will be the nucellus here. So this nucellus will remain even after the complete development of the seed, and such persistent nucellus will be called as what? Perisperm. Sorry, not the endosperm, it is the nucellus, the persistent nucellus that remains in some of the seeds even after the complete development, complete maturation of the embryo. Such a persistent remnant of the nucellus will be called as perisperm. This can be a very important 1 mark question. It can be a very important CET or NEET question also. I repeat, perisperm is the persistent remnant of nucellus. So in some seeds such as pepper, beet, etc. So I hope this is clear. So this is regarding the seed, the development of the seed, different parts of the seed, types of seeds such as albuminous and non-albuminous, and also regarding the perisperm. Moving on to the development of the seed. So now let us look at how exactly the development of the seed takes place. See, you know very well, this is the structure of the what? This is the structure of the ovule. Ovule, that is consisting of what? A completely mature embryo sac or female gametophyte, we call it as 7-celled 8-nucleated condition. Usually in the case of the angiosperms, it is also called as a polygonum type of embryo sac. This we have already studied. And in this case, now what happens? After fertilization, after the double fertilization takes place, after the male gamete fuses with the egg and the other male gamete fuses with the central nuclei. So what happens now is that, so the central structure or the embryo sac will develop into seed, and these two integuments, the outer coverings of the ovule, they will develop into, these integuments will develop into the seed coats. So usually in dicot seeds, there are two seed coats. Outer seed coat will be called as, see, you can see, look at this diagram, the outer seed coat will be called as testa, and the inner seed coat will be called as tegmen. Testa and tegmen. So these are nothing but actually what? Testa is derived from the outer integument. Tegmen is derived from the inner integument. And the actual seed is nothing but what? The embryo sac or the female gametophyte. And see, as I told you, in between the embryo sac and the integument, this space is completely filled by what? It is filled by nucellus. And this nucellus, in most of the cases, it deteriorates. That means it will degenerate. But only in some seeds, as I told you, in pepper, in beet. So this nucellus remains all along the life cycle. And such remnant of the nucellus is what is called as perisperm, that we have come across already, right? So, but this micropyle, as you see, micropyle is the point. It's a small pore through which the pollen tube enters into the ovule to cause the pollination, to cause the fertilization, right? So even after this entire ovule develops into the seed, the micropyle still remains. Through this micropyle, actually micropyle helps in the aeration, that means supply of oxygen or release of different gases, or during the germination of the seed, what happens? The embryo sprouts through that small pore called as micropyle. So like this, micropyle will still remain even after the complete development into the seed. So now let's go to the next this thing, pericarp. See, not only the fruit, as I told you, not only the ovule that matures to be the seed, but also the entire ovary, the lowermost portion of the gynoecium, that is ovary, that entirely converts into the fruit, right? So that fruit, it may be true fruit or false fruit. False fruits and true fruit, there are two different types of fruits. If a fruit that is developing from the ovary, the entire ovary, it develops or it matures into the fruit, then it will be called as a true fruit. In some cases, in some plants such as apple it can be, or strawberry it can be, or you must have seen, you have seen the fruits of cashew. So cashew fruits, usually they are like this. Cashew fruits are like this. So I hope you have seen, and the seed hangs below like this. So this is the fruit of a cashew. So you may wonder how the fruit is separate and the seed is outside the fruit. So as per our understanding, as we see in most cases of the angiosperms, seed will be present inside the fruit. But in the case of the cashew, we see seed is present outside the fruit. This is the fruit. This is because it is not a true fruit. It is actually a false fruit. So how this false fruit develops? So this means why it is called as false fruit. This fruit, what is that, juicy part or puffy part which we actually consume as fruit is nothing but the lowermost portion of the this thing, flower, that is what is called as thalamus. Thalamus in most cases will develop into the fruit. So not only the ovary, along with the ovary, a part of the thalamus also develops into the fruit, and such fruits which develop from any other part of the flower apart from the ovary, that is what is called as false fruits. So examples of false fruits can be apple, strawberry, cashew, etc., etc. I hope you have understood the difference between the true fruit and false fruit, right? True fruit means it develops only from the ovary, and it can be any type of fruit. So these fruits, angiospermic fruits, can be usually classified, or we can identify different parts on the different portions of the fruit, such as the outermost. See, this is the apple. I have taken a cross section of the apple, and here we can see the outermost coat will be called as epicarp. So carp stands for fruit, epi stands for above, mesocarp, middle region, and endocarp. Usually in most of the fruits, endocarp will be marked by the presence of the seeds. So here also we will see epicarp, mesocarp, and endocarp. So in most of the fruits of the angiosperms, we will see these three regions. And all these three regions, epicarp, mesocarp, and endocarp, all these three layers are collectively called as pericarp. So they are collectively called as pericarp. So this is the covering of the fruit. Entire fruit will be covered, and this entire covering of the fruit will be called as pericarp, which will be classified into epicarp, outer epicarp, inner or middle mesocarp, and inner endocarp. So like this. So like this, we come across different types of fruits, false fruits and true fruits, and different layers of the fruit. Development of the seed and fruit we have seen. Now coming to one more interesting aspect in this actually, that is called parthenocarpic fruits. You definitely have heard about the seedless fruits. You have heard about seedless grapes. You have heard about seedless papaya. You have heard about seedless banana. And what exactly are these seedless fruits? Seedless fruits, actually, technically, they have to be called as parthenocarpic fruits. So you can see the spelling here. Parthenocarpy means, parthenocarpy is a technique of production of fruits without fertilization. Not to get confused, be very clear, parthenogenesis is different, parthenocarpy is different. Parthenocarpy is related to the plants, development of the fruits in plants without fertilization. Parthenogenesis means it is a development of the young one, in the case of animals, directly developed from the female gamete. Right? So that is different. And here we are talking about parthenocarpic fruits. Parthenocarpic fruits, we can take the example of banana, we can take the example of papaya, we can take the example of grape. How it is done? In some cases, in some plants, so naturally the fruits will be developed without fertilization because of the hormonal variations. And in most of the fruits which are actually commercially utilized, see, when there is a huge demand in the market for banana or papaya, so what do? Is they spray some chemicals and you have studied different plant hormones, right? So you have studied about the auxins, you have studied about the ethylene. Ethylene is the fruit ripening hormone. It's a gaseous hormone which helps in the ripening of the fruits. So they spray this ethylene on unripened fruits of banana or papaya, and they make the fruit, unripened fruit, to get ripened. So that is how the fruits are developed, and such fruits are called as parthenocarpic fruits. So this is not the usual process of fertilization that is taking place. When there is no fertilization, obviously seed production will not be there, and that is why these parthenocarpic fruits, they are commonly called as seedless fruits. So I hope you understand why they have to be called, the common reason why they have to be called as seedless is because so they do not follow the normal mode of reproduction. They will be made to get ripened because of spraying of some chemicals. That is why as there is no reproduction, seed production will not be there, seed setting will not be there. That is why they are referred to as seedless fruits. Now, see, let us see a few diagrams of the seed. See here, we can see this is the detailed diagram. This is a detailed diagram. This is a mature embryo we can see here, and this is of course, this is also the similar diagram here. Here falls the micropyle. Micropyle falls here, and the development of the embryo takes place, and this is the embryonal axis. This central axis will be called as the embryonal axis, and embryo develops. So you can see SAM means shoot apical meristem. The lowermost portion is the root apical meristem. So shoot apical meristem can be covered by what? Epicotyle. Root apical meristem will be covered by coleorhiza, right? So like that. And so this develops into shoot, and this develops into root, and surrounding this there will be the endosperm. So this region, you can see this red region, that is nothing but the endosperm, and outside the endosperm you can see two seed coats. Normally in the case of angiosperms there are two seed coats. So in most dicots there are two seed coats. In the angiosperm monocotyledons also there are two seed coats. In some cases of the monocots, these two seed coats, they are joined with each other, they are clubbed, right? So that is a different issue. We can discuss that aspect in the CET, right? So this is the entire structure of the embryo, the endosperm and the seed. So no matter whether it is a dicot seed or monocot seed, so it is somewhat the same. Now, advantages of seed production for angiosperms. See, you know that seeds are produced only in two divisions of the plant kingdom, that is the gymnosperms and the angiosperms. Of course, gymnosperms also have got the wide range of applications or advantages of the seed productions as well as the angiosperms. So what are those advantages? The first advantage is that, so there is no need. Unlike the lower plants, these higher plants such as angiosperms, they do not need water for the dispersal of the male gamete to reach the female gamete, or they do not need water for pollination or fertilization events. So seed can be produced very easily by following a very easy mode of reproduction when compared to the lower plants. That is why seed is highly dependable. That means these angiosperm plants, they are highly dependable, they can be dependable on the seed for the production of next generation, for the next progeny to be produced, right? So seeds are highly dependable in the case of the angiosperms. That is what the first point talks about. Second says, seed dispersal mechanism favors, seed dispersal mechanism of the angiosperm favors the distribution of the angiosperms throughout the world on almost all the different vegetations, or on almost all the different types of environmental conditions in the case of on the earth. And that is why we say, see, angiosperms are said to be cosmopolitan in distribution. They are found in almost each and every geographical location of the world, of the earth. So be it in Arctic conditions, be it in the case of the desert or in temperate zones, angiosperms are present. It is because of powerful seed dispersal mechanism. And this seed dispersal mechanism is also associated with, it is also helped by one more or two more benefits or advantages. One is these seeds, especially dicot seeds, so they have got a large storage space because of the two bulged, cotyledons. There are two bulged cotyledons which will store large amount of food material, and this large amount of food material will help the seed to maintain viability. Viability means, so once when the seed is dispersed from a fruit, so either mechanically or by naturally, when a seed is released by a plant body from a fruit, so once again it has to go and meet the favorable conditions or moisture level, required moisture level, soil, so everything should be favorable. Only then the seed will, germinate. So from the release of the seed until it germinates again, so during this condition the embryo should not die. In order to keep the embryo in the healthy condition, so nutrition is required and huge amount of stored food material is required. That huge amount of stored food material, it is there, that is stored in the cotyledons of the seeds. So this is one more benefit, one more advantage of for angiosperms by the seed. So this also helps greatly for seed dispersal mechanism. So seeds can be dispersed for a very long distances as well. And there is one more benefit, hard seed coat, which will provide a protection. You might have seen many seeds, such as the seeds of sapota. Sapota seeds are very hard. You might have seen the seeds of which one? Or any seed. For example, the lemon seeds, or you have seen the seeds of mango, which are all very hard. You cannot destroy these seeds very easily because of the hard seed coat. The outer seed coat called testa, that is very hard and it cannot be destroyed very easily. It will provide the protection from mechanical injuries. It will provide the protection from high temperature or cold temperatures. It will provide the protection against various enzymatic activities or chemical reactions, right? And that is why during the journey of the seed, during its dispersal, seed will be protected, okay, very carefully, and it will once again help to maintain the viability and to germinate, right? So that is one more benefit. And the most important, when we talk about the variations, when we talk about the evolutionary aspect of the angiosperms are greatly helped by the mechanism of seed setting. That is because, see, you know that seed is nothing but what? It is the ultimate product of sexual reproduction. So sexual reproduction obviously involves the participation of male and female gametes. So the genetic material of male and female gametes, so its seed is nothing but it's a combination of the genetic material of male and female gametes. So this will bring about a new genetic combination, and when it sprouts to produce the next progeny, that will have some new genetic characteristics. This is what is called as variation. So we have discussed about variation in the previous classes also. So variation means a small genetic change from the parents in the next offspring. So that is what variations in the long run, they are going to bring about the evolution, right? So that is about variations. And these are all some advantages of seed for angiosperms. There is one more very important, what is that advantage for the angiosperms by the seed, that is seed dormancy. So you have seen the farmers storing the seeds for a long duration in gunny bags. So you might have seen the farmers storing the rice seedlings or wheat seedlings or groundnut seedlings or green gram seedlings. So various such seedlings are stored usually in the agricultural practices by the farmers. And you have seen that before these seeds are stored, they make sure that these seeds are completely dried up. So drying up of the seed is very important. So what is this? How it is associated with the topic that we are going to discuss about seed dormancy? See, seed dormancy, it is very simple. You have come across this even in the last chapter of the first year, that is nothing but plant growth and development. Seed dormancy means it is a sleeping condition of the seed or a resting condition of the seed. See, when the seed is mature, after the complete maturation of the seed, so seed is going to lose its moisture, and so 10 to 15 degrees, 15% of the moisture when it is lost, now the seed becomes dry and it persists to be alive in condition. And this living condition of the seed, even during the resting condition, that is what is called as viability of the seed. So in order to make it easy, see, seed viability means it is the capacity of the seed to remain viable or to remain living in condition That means it will be able to germinate once again when it comes in contact with the water and the soil. When favorable conditions come, so the seeds are going to germinate once again. So, but only during the resting condition. Seeds, they remain inactive. They remain dormant. They are sleeping seeds. But still, they have the capacity to germinate once when the favorable conditions arrive. So that is what is called as viability. And this dormancy has helped the seeds to propagate the angiosperms again cosmopolitanly. So because why it happens like this is when the seeds are released by the fruit. So during the dispersal of the seed, during their journey of the dispersal, so they may come in contact with the favorable conditions very soon, or it may take forever, it may take very long time. So, and this also has helped the agricultural practices greatly, because farmers, when they take the yield this year, so they will store the seedlings of this yield for the next year. For 1 year, these seeds can be stored without any harm. So, seed dormancy is a resting period or sleeping condition of the seed where viability of the seed is still maintained. It is still persistent. So, for how long? Some species of the angiosperms may be having the seed viability for few weeks to few months. Some may be having some years. And in some cases, these are the extreme cases which have been discovered here. Lupinus arcticus, a plant belonging to the family Fabaceae, Lupinus arcticus. It was discovered during the excavation in the Arctic region, Arctic tundra. So, it was found in a very extreme cold condition or cold weather atmosphere. So, it was estimated to having 10,000 years of dormancy. That means the seed was still viable. It was still in living condition after 10,000 years. So, seed was in dormant condition for 10,000 years. When it was taken out of dormancy, when they tried to sow the seed, it germinated even after 10,000. So, by using different techniques such as carbon dating, the age of the seed was estimated. One more recent example that we can see is that of Phoenix dactylifera. Phoenix dactylifera is nothing but Kharjura, what we call it in Kannada. So, Kharjura, the seed of Kharjura, Phoenix dactylifera, so was found to be 2000 years old. That means it was, the viability of that seed was, or dormancy period of that seed was 2000 years old. This was also found during the archaeological or historical excavation of a palace by King Herod. So this palace was built by King Herod, and this palace was found near the Dead Sea. So near the Dead Sea this palace was found, and in that palace one seed of Phoenix dactylifera that was found to be 2000 years old, and it was found to be still viable. It had the capacity to germinate once again. So this is seed dormancy is yet again one more very important tool, or one more very important advantage of the propagation, for the propagation of the angiosperms. Right? So this is about the seed. So I just repeat in a nutshell. Seed, it will contain seed coat, cotyledons and embryonal axis. So depending upon whether it is a monocot or dicot, cotyledon may be 1 or 2. There are different types of seeds, albuminous and non-albuminous seeds. Albuminous means residual endosperm is present even after the complete development of the embryo. Example, wheat, maize, barley, sunflower, castor. And non-albuminous seeds, they do not have any residual endosperm after the complete development of the embryo. So example, pea and groundnut. Perisperm, it is the persistent nucellus that is present even after the complete development of the embryo. That is what is called as perisperm, present in pepper and beet. So other aspects, seed development, how does it take place? Fruit development, false fruits and true fruits we have seen. Parthenocarpic fruit, development of the parthenocarpic fruit naturally or artificially by making use of different plant hormones. So, and then we have discussed about advantages of seed setting or seed production in angiosperms. So this is regarding today's session.